Now we're going to jump right into our first session, which is clinical trial updates with presentations from Dr. Jin of Novel Pharma and Dr. Shin of GC Biopharma, sharing updates on their ICV enzyme delivery for Sanfilippo syndrome type A. We're going to then be joined by Dr. LeWinter from JCR Pharmaceuticals, who will share updates on JCR's Sanfilippo Type A and Type B enzyme replacement programs, and then that will be followed by a Q&A. So I will bring Dr. Jin and Dr. Shin up here.
Okay, thank you. Good morning and good evening. Thank you for inviting me to Sanfilippo Community Conference. My name is Sookyung Shin, and I'm the head of medical division at GC Biopharma. 2024 was and is truly a remarkable year for Sanfilippo community and GC Biopharma as well in many ways. I would like to thank organizers for giving us opportunity to talk about our project at this exciting event. Today's presentation is co-presented with Dr. Jin, who is the CSO of our partner, Nobel Pharma. Next, please. This is disclaimer. Next, please. GC1130A is an enzyme replacement therapy that is given through the injection into brain's ventricles. GC Biopharma and Nobel Pharma have collaborated to develop GC1130A. So far, GC1138 has been granted orphan drug designation from the FDA and EMA, as well as rare pediatric drug designation and fast track designation from the FDA. Additionally, GC1138 has received investigational new drug clearance from US, Japan, and Korea. Next, please. Let me briefly introduce our collaborator, Nobel Pharma. They are focused on developing treatments for rare disease, rare pediatric LSD and CNS diseases. Dr. Jin, who is presenting together with me today, is the company's CSO, and he has extensive experience in treating lysosomal disease. He had led clinical trials for treatment such as idursulfase beta IV for Hunter syndrome, and had also worked on preclinical studies for ICV ERT treatments. Currently, experienced team in Nobel Pharma, including Dr. Jin, is collaborating with GC Biopharma on the GC1130A project. Next, please. GC Biopharma is a Korean company with over 50 years experience. We specialized in biologics such as vaccine, plasma proteins, recombinant proteins for rare disease, covering the entire process from research to manufacturing. Our biologics are manufactured in 3 GMP sites in Korea. We are expanding our presence worldwide and have recently received US market approval for our human immunoglobulin product, which is Alliglo. GC Biopharma has developed Hunterase ICV for Hunter syndrome, and it is now available in several countries, including Japan. Today, I will be sharing our experience with ICV ERT for patients with Hunter syndrome, and Dr. Jin will update on the progress of GC1130A for Sanfilippo syndrome. Next, please. Mucopolysaccharidosis, MPS, is a genetic disorder caused by lysosomal enzyme deficiencies. There are 3 types of MPS, each depending on the specific enzyme affected. Heparan sulfate deposition is particularly associated with the brain deficit in MPS 1, 2, and MPS 3. Enzyme replacement therapy aims to provide the missing enzyme from outside the body. The lysosomal protein naturally has the ability to move from outside the cell to inside using mannose-6-phosphate receptor, and ERT drugs are also taken up by this receptor. Next, please. In the case of MPS 2 and MPS 3, undigested heparan sulfate is accumulated in the lysosome, causing it to enlarge and lose its function. This ultimately leads to brain damage, as shown in the MRI scans of MPS patients in the picture at the left side end. The brain lesion may not be apparent when the child is younger than 2 years old. The picture in the middle is the data from Dr. Sapiro's paper and shows us there is a critical period of time for treatment of MPS patients. Therefore, our goal is to replenish ERT enzyme to the brain of patients as soon as possible and as much as possible so that the patients do not miss the golden time. Conventional ERT administered intravenously cannot cross the blood-brain barrier. However, our mouse study in the right side picture showed the ICV administration effectively delivered the enzyme to the brain. Next, please. In 2021, idursulfase beta for ICV administration was approved in Japan. CNS symptoms are one of the medical unmet need for MPS II patients. Once the ICV device implanted, patients can do daily activities such as bathing and swimming. MPS II patients received 30 mg of enzyme once a month. The overall injection time is 1 to 2 minutes. Next, please. Last year, we released safety and efficacy follow-up data for idursulfase beta ICV for up to 5 years. The data indicated that idursulfase beta ICV was well tolerated and safe. 50% of the patients experienced mild adverse event related to the reservoir, all of which were resolved with proper treatment. Common drug-related adverse events included vomiting, fever, and nausea. Next, please. As our patients have ICV reservoir implanted, we could collect CSF sample at every injection before dosing and closely measure heparan sulfate CSF levels. Patient maintained low CSF heparan sulfate during follow-up. In the right picture, you can see 2 out of 6 patients continued to show development age gains, and 2 were stabilized above natural history line in DASH. With our knowledge and experience on ICV ERT development from Hunter syndrome, we are confident that we can bring treatment for MPS III patients as well. Moving forward, Dr. Jin will share the non-clinical study result and our phase 1 clinical study update for GC1138 ICV ERT. Dr. Jin, over to you.
Yes. Dr. Shin, thank you so much for your taking effort and introduce me. Whereas, next please. Yeah. Whereas, from now on, I take over this presentation and we'll start from the animal study efficacy data. This is a data from the mouse brain of the protein that we investigational products when we intracerebroventricularly administered this drug to the brain. By the way, Dr. Shin introduced. Then you can see in the left side there is ICV high dose, low dose, and IV high dose graph. You can see there is a very huge difference from the IV to ICV, like exponential curve. And also in the right side, there is in the left side it was a CSF, but in the right side it is a brain itself measured from the brain and directly. Then you can see the IV administered drug is not measured because it is below the detection level, and only the ICV administered, and you can see there is a very stable, stable remaining drugs in the brain itself. This is very interesting and important. Next, please. Then we can compare between, there is two types of delivery method directly into the CS. One is our method ICV, and the other is intracerebral way. You can see in the left side, there in the CSF, when we administer this drug to IV, ICV, and intrathecally, then between the ICV and intrathecally administered, there is not much difference in the CSF. However, in the right side, you can see brain distribution of these drugs to ICV versus IT shows the big difference. In the upper panel, there is an ICV administered one, and the lower one is intrathecally administered. In the intrathecally administered, most of the drug will remains in the spinal cord. You can see in the right side, IT, the spinal cord. However, if we administer this via ICV, then you can see the drug still remains in olfactory bulb and cerebral cortex, and everywhere, quite even all across the brain. So this shows the strength of ICV way over IT. Next, please. This is biochemical efficacy of ICV administered this drug. You can see in the brain and in the CSF also there is a decrease in the heparan sulfate in a dose-dependent way. Shows that this drug very effectively can reduce the heparan sulfate in the brain, even in one single injection. Next, please. So this is a new data that we just had, and you can see that it's a behavioral data from the MPS IIIA mice. In the left side, you can see that in the wild type mouse, it's very busy going around, so there is many lines. But in the right side, MPS IIIA knockout mouse, there is very sparse footprint. But when we administer just one single ICV injection of this drug, this mouse will regain their vigilance, and it can have a very similar walk pattern like wild type. Also, the hind limb rearing and the total activity of the mouse will improve after ICV single injection. Next, please. So now I will show you the clinical part of our trial for the phase 1. You know, the drug IP is a recombinant human and heparan and sulfatase, and you can see it is administered into the human brain will be administered in human brain intracerebroventricular. Yeah. Okay. See. Next, please. For this phase 1 clinical trial, we have 3 countries registered, 3 sites in United States soil, and 2 sites in South Korea, and 1 in Tokyo. So it is a multinational study, and we can say that in Oakland Clinic, we will have our first patients. Next, please. This is just a summary and just a brief explanation of our clinical trial phase 1. And you can see there is in the middle part, dose escalation period. There is a consist of 3 cohort, cohort 1, 2, 3. It goes up with every dose, every escalation dose, and each cohort will last 8 weeks. After first cohort is finished, then if there is no problem, and after review from the safety committee, then move to cohort 2. Then after it is over and the safety is okay, then move to cohort 3. And you can see in the right side, up to 9 patients can be enrolled in this study. And we can say that we are now in the middle of the screening of the first patient, and we'll have our study in November, start from the November, we can say that. So we are very excited about this clinical trial. Okay, thank you so much.
Thank you, everyone. Thank you, Glenn and Kara, for this very kind invitation, and the Sanfilippo community for hosting this collaborative conversation and sharing some very personal stories yesterday. My name is Robin LeWinter, and I am the JCR USA Medical Affairs and Medical Communications Lead. I am here with my colleague, Angelica Cordoba, who leads our clinical programs in the Sanfilippo syndrome, and she'll be available to answer any questions during the Q&A session. I'm looking forward to sharing with JCR who JCR is and discussing our exciting platform designed to deliver enzyme across the blood-brain barrier and to provide new treatment options for patients with MPS. Next slide, please. Here is our forward-looking statement. Just leave it up for 1 second so you can read it. Next slide, please. Before I get into details about our platform and therapy pipeline focused on Sanfilippo, please allow me to introduce my company. Next slide, please. This slide presents JCR history. I understand this is a very busy slide. There's a lot on this slide, so I'm only going to go over some very key points. JCR is a Japanese pharmaceutical company founded in 1975. Its 49 years of history highlight its growth and focus on research and development of process technologies and new treatment options, especially for orphan and rare diseases. Since JCR's founding almost 50 years ago, it has been a regular driver of innovation, until recently predominantly in the Japanese market. After initially developing drugs using established purification technologies, JCR led innovations ranging from cell development to cell culture technologies, including the first allogeneic regenerative medicine product in Japan. More recently, JCR has established a platform technology called J-Brain Cargo that enables molecules of interest to penetrate the blood-brain barrier. We are proud that this has resulted in the successful approval and launch of an enzyme replacement treatment for MPS II in Japan in 2021, and a global phase 3 clinical study is ongoing in countries outside of Japan. Next slide, please. As I mentioned, until recently, JCR was a domestic company. You can see here that the headquarters are in Kobe, Japan, with a number of other offices distributed in the rest of Japan. Research, development, and manufacturing are very important activities for JCR. Out of the 1,000 employees today, about 35% are focused in R&D and 30% in manufacturing. 2 research centers focused on the discovery and proof of concept of innovative technologies and products are located in Kobe. There are 5 manufacturing sites in the neighborhood that sustain our products marketed in Japan and in clinical trial products for global studies. As we continue to explore the potential of the game-changing J-Brain Cargo platform technology for the treatment of rare diseases, JCR has expanded our global footprint and is now reaching out to patients beyond Japan. Today, we have a presence in the USA, Brazil, and the Netherlands to sustain global clinical development in our programs for rare diseases. Next slide, please. I'm now going to share the basic premise of J-Brain Cargo technology and why we are so excited to explore its potential in mucopolysaccharidoses, including Sanfilippo syndrome. Next slide, please. Now, I probably don't need to get into much detail for the people on this call, as the audience is likely well aware, but just to remind everyone, MPS is a lysosomal storage disorder. Healthy individuals are able to break down large sugars in the lysosomes of their cells using various specialized enzymes. In a patient with MPS, there's a mutation in one of their genes which causes a faulty enzyme. If the enzyme is dysfunctional, the lysosome is unable to break down the large sugars like heparan sulfate or dermatan sulfate, and these accumulate in the lysosome and eventually damage the cell. These sugar macromolecules are distributed all over the body, so the manifestations of lysosomal storage disorder leads to disease burden distributed throughout the body, including somatic organs, bones, eyes, and the central nervous system. And this is measured by extreme elevations in heparan sulfate in various parts of the body. Next slide, please. So once it was discovered that the underlying pathology in MPS disorders was these missing enzymes, a simple solution seemed to be exogenous enzyme replacement therapy, or essentially providing the missing enzyme to patients. While enzyme replacement therapy has been shown to provide benefit to somatic organs, it is unable to cross the blood-brain barrier. So what is the blood-brain barrier? It's a semi-permeable membrane that protects the brain and central nervous system from harmful substances. It is essentially a very selective filter that blocks the passage of many substances from the blood to the brain. Why do we have it? It's pretty simple. It blocks a lot of pathogens from entering the brain, and you definitely don't want germs infecting your brain. Unfortunately, it also prevents exogenously administered enzymes from entering the brain. So while exogenous enzyme replacement therapy treatment provides benefit to somatic organs, it is not beneficial to the brain. Next slide, please. The J-brain cargo technology is designed to take advantage of the body's carefully regulated blood-brain barrier to deliver enzyme to the brain. So how does this work? Well, all cells need iron for oxygen exchange, and iron is safely carried around the body by a specialized glycoprotein called transferrin. As the cells in the brain need iron to function as well as in the body, your body has created a mechanism to transport transferrin across the blood-brain barrier. Transferrin, as indicated by this little red dot over here, binds to a specialized receptor, cleverly named the transferrin receptor, on one side of the blood-brain barrier. It is encapsulated and then ferried to the other side of the blood-brain barrier, where it is released into the parenchyma in the brain and is now available for use by the cells. J-brain cargo has hijacked this process. We've developed a technique to fuse or connect an antibody to the transferrin receptor to an enzyme. So if you remember from your biology, an antibody is like a puzzle piece which can connect to its antigen. In this case, the transferrin receptor is the antigen, and it exactly matches the transferrin receptor antibody on the J-brain cargo. And just like it does with transferrin, the transferrin receptor binds to the antibody in our drug product, encapsulates the whole drug product, and carries it across the blood-brain barrier, releasing it into the parenchyma in the brain. It is key to point out that the missing enzyme is attached to the antibody, so the missing enzyme is also ferried across the blood-brain barrier and released into the parenchyma. So while our drugs are administered intravenously and into the blood and distributed throughout the body, they are able to take advantage of hijacking the transferrin receptor to cross to the blood-brain barrier. And we are so proud that this platform has led to the development and approval of IIIA in Japan in 2021, which has been shown in clinical trials to reduce the production of heparan sulfate in the brains of patients with MPS II. Next slide, please. So we've discussed that J-brain cargo is able to cross the blood-brain barrier and enter the brain parenchyma. It's also important to note that since it's administered intravenously, it is readily available for uptake in all somatic organs via the enzyme receptor, the mannose-6-phosphate receptor, and transferrin receptors on all cells, both in the CNS and the somatic organs. Therefore, we postulate that this treatment provides benefit both in the central nervous system, but also on somatic signs and symptoms. And a reminder that MPS does affect many organs in the brain. So next slide, please. JCR is continuing to expand on its success with MPS II, and we're developing treatments for other MPS syndromes. I'd like to share with you data supportive of JCR's treatments for Sanfilippo syndromes. These have been confirmed in animal models of the disease, and we have or are very soon to kick off early stage clinical trials. Now, our treatment for MPS IIIA is currently very creatively called JR-441, and we have performed proof of concept studies in animal models of MPS IIIA. Next slide, please. In order to perform these proof of concept studies, we engineered mice in two ways. First, we've created a mouse model of MPS IIIA by knocking out or removing the SGSH gene from the mice, which mimics the mutation in MPS IIIA patients. We've also added in a gene that allows the mouse to express the human transferrin receptor, so it is able to detect the human transferrin receptor antibody in J-brain cargo. We then intravenously infuse the mice with JR-441, an antibody enzyme fusion protein specific for transferrin receptor and fused to SGSH enzyme. To confirm the drug is working as expected, we measure the substrate. In this case, we measured heparan sulfate, and a reminder that heparan sulfate is one of the macromolecular sugars that cannot be broken down in patients with MPS IIIA. If JR-441 is working, we would expect there to be less heparan sulfate in the brains of mice with MPS IIIA. So next slide, please. So this is a really busy and complicated slide. I promise it is the busiest one that you're going to see today. So please be patient with me as I talk through it. I would like you to first focus on the graph in the middle, top middle, called serum or blood. And on the left, on the very left of this graph in the middle, we see levels of heparan sulfate in the blood of the normal mouse. And as expected, in a normal mouse, levels are low. If you look next to it, the black bar, this is the diseased mouse. And here we can see that the level of heparan sulfate in the blood of a diseased mouse are much, much higher. Now, if you look at the three bars to the right of that, called low, medium, and high, this is a diseased mouse that is treated with JR-441 for 13 weeks. And what you can see here is that the dose as JR-441 dose increases, the amount of heparan sulfate in the blood decreases to near normal levels. Now, if you look all the way on the right on that one graph, you can see a diseased mouse that has been infused with SGSH enzyme replacement therapy alone, not the J-brain cargo. And you will note in this mouse, you'll also see a decrease in heparan sulfate, which tells us that enzyme replacement is able to reduce the levels of the substrate in the blood and the organs, whether it's delivered from SGSH enzyme replacement therapy alone or from J-brain cargo. But now I'd like you to look at the graph on the top left, the one that says brain. This shows us the heparan sulfate levels in the brain. On the very left, you'll see that heparan sulfate levels in the normal mouse is very, very low. But if you look to the right of that in that dark black bar, you will see that levels in the diseased mouse is very high. Once again, if you look to the three little graphs after that called low, medium, and high, you'll see that after infusion with JR-441 in the diseased mouse, heparan sulfate levels dose-dependently decrease in the mouse brain. Now last, I'd like you to look at the red square. This is a diseased mouse who received exogenous SGSH enzyme replacement therapy alone, not JR-441. Now remember, the brain is protected by the blood-brain barrier, and normal enzyme replacement therapy cannot cross the blood-brain barrier. You note that the SGSH enzyme replacement therapy alone is unable to cross the blood-brain barrier and decrease heparan sulfate levels, while JR-441 crosses the blood-brain barrier and decrease heparan sulfate levels in the brain. So this slide shows similar results in the CSF, or the cerebral spinal fluid, which also requires the drug to cross the blood-brain barrier, while other organs in the body, the heart, kidney, and liver that are not protected by the blood-brain barrier, do benefit from both JR-441 and SGSH enzyme replacement therapy alone. Next slide, please. This This slide provides additional evidence where we're looking at stains of brains of mice that have been treated with our drug. So if you look on the left, this is the healthy, this is a healthy mouse, and these are the Purkinje cells in the cerebellum. And if you look at the middle cell, this is a sick mouse, and you can see the little red arrows point to dark brown cells. These dark brown cells are staining for CD68, which is a marker of microglial activation, which is indicative of diseased and dying cells and brain damage. Now, if you look at the brain on the right, this is a diseased mouse that has been treated with JR-441, and there are no dark brown cells, which is suggesting that JR-441 treatment suppresses microglial activation and preserves brain cells. We have initiated a phase 1/2 clinical pilot study in human patients in MPS IIIA in Germany, and details of this study can be found on clinicaltrials.gov. Can I have the next slide, please? Now, I'd also like to briefly share an overview of JR-446, which we're working on for the treatment of MPS IIIB. So next slide, please. Just like with 441, with 446, we've created a mouse model of MPS IIIB by knocking out the NAGLU gene, which is the mutated enzyme in the MPS IIIB patients, and it has the ability to express human transferrin receptor. We've infused these diseased mice with JR-446 again, JR-446, and again, we measure heparan sulfate levels. Next slide, please. So this slide is now slightly less busy than the one I showed you with JR-441, but still a little busy. Let's look at the top left. You'll see that in the normal mouse has very low levels of heparan sulfate in the brain, while the diseased mouse has very high levels in the brain. Increasing doses of JR-446 over a 4-week period decreases the heparan sulfate levels. We observe this same effect in the serum and the heart, suggesting the benefits are observed both in the CNS and the somatic organs. Next slide, please. Finally, staining of the brains of these mice with MPS, a model of MPS IIIB. We've stained the mouse brains of these mice, and we've looked at the Purkinje cells in the cerebellum for microglial activation. And here we're looking at LAMP1 staining. You can see many dark brown cells in the diseased mouse brain, which has been drastically reduced in the treated MPS IIIB mice. And again, we have a phase 1/2 clinical pilot study in human patients that will soon start enrolling in Japan, and details of this can also be found on clinicaltrials.gov. Thank you for letting me share the exciting work we are doing at JCR, advancing treatments in Sanfilippo syndrome, and will be available during the Q&A session. Oh, this is our pipeline, but this is just for backup.
Good morning, good evening, everybody. Thanks for the wonderful presentations. Our clinical trial updates are obviously, of great interest to everybody in the community, but especially the patients and families that are out there waiting. So appreciate you sharing those updates. We do have some questions for the group, and we'll start out with maybe some location questions. For the novel GC team, are the 3A trials being conducted? I'm sorry. We're also welcoming Dr. Angela Cordoba from the JCR team. So welcome, Dr. Cordoba. You mentioned the 3A trials being conducted in Korea, and actually, I think you answered that. There are a couple of sites. Is that right? 2 sites in South Korea?
There are 2 sites. Yeah, there are 2 sites.
And when will they begin enrolling? Also November?
November, end of November or early December. But however, we have checked the patient for the first patient should be checked for 2 months. So we have to wait for 2 months at least after the first patient is enrolled. So the timing can be different a little bit.
Okay, depending on where you enroll the first patient.
Okay. Right. Yeah.
Understand. And then there's questions on whether you plan to also initiate or move forward with a 3B program as well for your ICV approach.
As of now, GC Biopharma do not have the plan for the MPS 3B yet. And Dr. Jin, do you have any plan for 3B?
We are the same team.
Okay.
We don't have a plan to.
Okay.
Okay, thank you. And then for the JCR team, families were also wanting to know if you had any plans for a trial site in Korea for your 3B intravenous enzyme replacement program, or if you could share any more information on your clinical trial that is in the works for 3B.
Should I take that one, Robin? Robin? Oh, sorry.
I think she might be frozen.
Yeah, okay. Yeah, no, I can respond. The current trial, as you know, and you can see in clinicaltrials.gov, is only in Japan. Of course, we are planning to have the next phases of development, a phase 2-3, but right now is still under discussion and we haven't selected any countries yet.
Okay, thank you. And then do you have any information on when you might anticipate being able to share any clinical data from your pilot phase 1-2 study on the Type A program?
Not yet. We are continuing with the protocol as it is, but we don't have yet the plans. Yeah.
Okay. Back to the novel GC team. There were some questions about the mouse data, and so thank you for sharing that additional behavioral data. We've seen in the literature sometimes the MPS 3A mouse and patients are described as hyperactive or having increased activity, but your results show lower activity levels in the open field. And there's questions about, maybe you could, give us some insight into that, if there are any differences between the particular mouse model you used, the knockout versus other mouse models. And then I have a follow-up question after that.
Okay, so I understand your question, and actually, our team, Novel Pharma, our team has an excellent PhD who deals with these kinds of animal model. So I just asked her, and she said that first, it is naturally occurring the mouse for the 3A model, and that is one answer. And the other answer is that there is another question, and it was what was the age of the knockout mouse, and that was another question. And I also asked her, and she told me that from the 4 weeks of a mouse, they manifest some of the symptoms of the Sanfilippo mouse. And for these experiments, we used 6 weeks of age of a mouse for this experiment. Is it enough for answer?
We cannot hear you.
Oh, I'm sorry, I was muted. Yes, thank you very much. Yes, I understand that the open field data you shared was from 6-week-old mice, and they were naturally occurring mice, not knockout mice.
Yeah.
Okay.
Yeah.
Got it. Okay, back over to Dr. LeWinter and Cordoba. Could you comment on the frequency of the injections used in the JR-441 mouse study?
Yeah, I apologize. I got kicked off. Oh, I was having some connection issues, so I'm sorry if I missed any questions for me. We at this point, we cannot comment on the concentrations used in the mouse model.
Okay. Understand. Okay. And let's see, did we catch everything? Dr. Jin, the GC1130A is a recombinant enzyme. And I wondered, when you showed the mouse model data showing that even when you gave it IV, intravenously, that there was some evidence of appearing in the CSF, though much, much lower than if you give it directly, CV. And so I guess I wondered, is the, your proprietary enzyme, is that at all different from native enzyme? And will we expect native enzyme, if given and unmodified, to be able to actually cross the blood-brain barrier, which is not the typical paradigm that we hear, but we're always learning new things. So I guess my question is, does your enzyme vary from native enzyme?
I think that there's a part we cannot reveal all of the details because it is related to our product. But we can say that it is not modified enzyme. Basically, it is not modified.
Okay.
Yeah, Dr. Kara, we are utilizing the ICV device to enter into the brain directly.
Understand. Yes. Yeah. Okay. Thank you. Thank you. I think we have a last question here for the JR-446 HSO4 levels in various tissues. What are they? So just seeking more information about the heparan sulfate levels in the different tissues.
Okay, that was my question. Is heparan sulfate, right?
I believe that's what I believe so.
Okay, yeah, I mean, we are assessing heparan sulfate, right? And so I guess that, yeah, that's the question. Yeah, I mean, we have assessed that in brain and all the somatic organs as well. So yeah, we have assessed that and we have seen the reduction.
Okay. Okay, just looking back over to make sure we've captured everything. In your different programs, do you find that the reduction level of heparan sulfate in the mouse models, are they getting, to that level of, just that critical level of detection, or do you find that they're normalized? Whatever normalized is for the mouse.
Yeah, that's the question. What is the normalization in the mouse, right? I mean, of course, we are seeing a dose reduction in a dose-dependent manner. And this is important for a dose selection that I can see that is actually one question that they are asking as well. I think that we are getting robust reduction as, Robin showing the screen as well. I don't know what should be the normal, but we are still able to assess, yeah.
Okay. All right. We have so many questions that I think maybe we'll have to take in follow-up after the end. And I would encourage anyone who might want to interact with our presenters. You can also use the chat feature and follow up with them as well. You're happy to do that. And thank you all so much for your time and your presentations today. We appreciate the work you're doing.
Thank you.
Thank you.